and welcome everyone to this webinar. We are very happy to have you here today. We see a lot of people, so that's really great. And today we are going to talk about generative AI under pressure, as mentioned by Leonora. How we can apply GenAI in grounded to reality, grounded in the constraints of what experts expect from this type of technologies. So first, we will focus on problems, we need to face in drug discovery and how we can use GenAI. Then we propose a different approach in GenAI compared to what has been published previously. We will show, again, several experiments how this GenAI is different, and also how can we apply in real use case this type of GenAI approaches and what type of output we can expect from it. And finally, we'll show you how we have integrated this technology in our software Machia, but a preview of Machia V2 with very nice and new user interface. So first problem, open problems of GenAI in drug discovery. One of the main difficulties when we talk about GenAI is that you need to add plenty of different type of constraints if you want to have a reasonable output. As we say here, successful molecules must meet multiple design goals, because having a good molecules, it's a very complex exercise. What main difficulties is that as much as you add constraint to the system, it's become very difficult to explore the chemical space. And usually what has been also highly described in the literatures, one of the byproducts of generative AI is that it tends to increase structural complexity during optimization. And this is what happened as well in medicinal chemistry. That's why such metrics like ligand efficiency have been introduced to avoid increasing complexity against activity, for instance. Which type of constraints are we talking about? We are talking about very simple physical descriptors, log-P, TPSA, PFI, QED, this type of easy to calculate physical descriptors. Then, of course, it's not enough. When you design molecules, you're targeting good predictive activity, good predicted solubility, toxicity, etc. So you will introduce QSAR models. On top of that, and depending where you are in your project, you can also introduce 3D scores, ligand-based, structure-based. Of course, chemists want to add their grain of salt, I would say, and they are also expecting some molecular features. They want to see some function. They want to see some presence of substructures, absence of substructures. So they also will add this type of constraint to the system. At the end of the day, all those constraints together, you want to have a molecules which is feasible. And not only feasible, but also you're targeting a synthetic strategy, meaning that you also want to have some key reaction type, key intermediate, maybe a list of providers. Maybe you want to have your inventory taken into account in that generative process. So as you can see, generating AI is not only about generating molecules, it's generating molecules against a lot of constraints. And again, the more constraints you add, the harder it is to explore the chemical space. Another very important problem and difficulties is how to balance feasibility and novelty. That's why during many years, people work in virtual screening, because virtual screening are pre-compute libraries where the chemistry is very well optimized, but still it comes at the price of low diversity, because 10 to the power of 10, it seems huge, but it's still very far from the 10 to the power of 60 theoretical molecules we can imagine. So the counter part of virtual screening is GenAI. First publication in 2016 was kind of a good hope, because at that moment we thought that the generative AI, the generation of molecules, was a solved problem. We could infinitely explore the chemical space. But we have been able to observe that this capacity of generating high diversity, novel scaffold, and explore the chemical space come with a price which is low feasibility, difficulties to make the molecules. So there is no point to generate the molecules which maximize plenty of different type of scores if you cannot make those molecules. And maybe somewhere we can imagine a world where we have the advantage of virtual screening, so a good synthetic feasibility, but at the same time, the advantage of high diversity and GenAI. And we are talking about synthetically aware GenAI. If we go to the literatures and if we have a look to what has been published, a very nice paper published in Nature in 2024 reports 43 methods published so far describing how we can use GenAI to imaging new molecules. If you go to this paper, you can see that in all methods, we have either graph-based GenAI, smile-based, fingerprints, selfies, different type of I would say, representation, which does not take into account synthetic feasibilities. All those type of molecular representation are not taken into account available building blocks, available synthetic transformation, and even synthetic feasibility. So at the end of the day, whatever the method here is, you will generate molecules without controlling synthetic feasibility. So you can rapidly be trapped in a digital space, I would say, and you will never be able to go to the lab. And I think, and this is a topic of today, and to present our GenAI at Iktos and what sets Iktos apart is that we have been able to develop a chemistry-driven, synthesis-aware generative design technology. Why others are struggling is because LLM know words, not chemistry. So if you talk about smiles-based generation, you're playing with natural language technologies on smiles or on selfies or on fingerprints, but it does not take into account the chemistry, the building blocks, the reactant, the reagents. So at the end of the day, you will maybe able to generate novel molecules like this one, or this one using this type of approach. But can you make these molecules? Maybe, but I can guarantee that this piece here will be very difficult to introduce. The character permutation playing with smiles doesn't guarantee at all synthetic feasibility. So this is where I think, you know, navigating into the space of characters permutation versus navigating to the space of ingredients, building blocks, and reaction provide a totally different type of outputs. And we'll be able to show you that soon. So at the end of the day, what chemists need from GenAI? Again, we have in one part virtual screening, which is very well-known technologies, which guarantees some level of success in terms of synthetic accessibility, but it's slow and expensive. If you have 10 billions of molecules in your library, you need to test all or to introduce some filters to expect at the end of the day to get something. What's cool with GenAI, it's like you have optimization. It's not a random selection. The system learned by the constraints to generate something which makes your goal. But again, it comes with a price, which is low synthetic accessibility. So the GenAI we have designed sets between those two worlds. We estimate currently that the space, chemical space we can navigate in is 10 to the power of 27. So a huge chemical space where we can guarantee to you that we can identify easy to make molecules in order to have a rapid DMTA cycle. Second, we can guarantee you that we can find and we can optimize those molecules into this chemical space to reach and to maximize several endpoints simultaneously, but also the novelty, it's very important. On top of that, again, chemists want more than feasible molecules, than optimized molecules. They want to be in control. they want to guide the system. They want to be able to pilot GenAI. They don't want to see molecules popping from nowhere, maximizing a certain set of constraints. They want to pilot that. And I will let Stephanie present. Stephanie, the floor is yours.
Yes, absolutely. So now I will take control back. So yes, so now the second module that we are going to discuss today is now we are going to talk about how we rethink GenAI for chemistry, and more specifically, how we enable chemistry-driven design strategy using our platform. So unlike typical GenAI models that just generate smiles or optimize black box scores, our GenAI is built to use chemical logic. So it understands structures, reactivity, and constraints at the building block levels. That means that we can ask chemically meaningful questions in the presence, for example, of a reference molecules like this one, to develop new ideas. And the kind of questions that we can ask as a chemist are like, for example, what if you replace the R1 moiety here with synthetically accessible alternatives? Or what if we substitute the head group R2 using specific name reactions? Or finally, what if we explore new heterocycles by hopping out the core under substructural constraints? So to answer the question if GenAI can use chemical logic to generate new ideas, the answer is of course yes. So specifically, our GenAI enables two key chemistry-driven design strategy. The first one is growing new molecules, and the second one is linking fragments into novel compounds. So in the growing mode, we start from a non-substructure and selectively expand it. In the linking mode, we bridge fragments to generate synthetically viable compounds. So here, there's a closer look at how it actually works. So on the left, our GenAI grows a molecule step by step using real reaction templates and respecting reactive constraints. So on the right, it links fragments by proposing realistic reaction-compatible connections, and it follows the same logic as the fragment growing that I showed you on the left side. So the important message here is that our GenAI is grounded in how chemists actually design molecules. This is where our technology really separates from standard GenAI. Every transformation you see here is grounded in reaction reality. So our GenAI does not hallucinate. It works from building blocks, applies validated reaction templates, and constructs synthetic pathways that are actually usable. So it's a generative system that respects synthesis by design. So if you want to have full details on our work, we lay them out in our preprint. So here we break down how the growing and linking optimizers work and how the models are trained and how the architectures actually work with chemical logic from reaction selection, building block selection, and also retrosynthesis stream. So now that we actually in the previous section, we analyze how GenAI can effectively use the in silico chemistry experimentation. The second point that also Quentin mentioned at the beginning of the presentation, what about the synthesizability? So for that, we need to review a little bit our algorithm and how it works. So as stated before, our GenAI does not just guess molecules. It builds them one step at a time with synthetic feasibility at every decision point. So our GenAI starts by searching 1 million building blocks using AI-guided prioritization. So it picks the best one to begin with a synthesis. So in this specific case, it's a protected spiral amide. Then it predicts the most suitable reaction type from over 2,000 organic transformation, predicting the optimal transformation. So that first building block and reaction yields a viable intermediates. So in this case, it's a bug to protection to unlock a key amine. Now the process repeats. Our technology is able to go through to another round of selection of building blocks and reaction templates based on these created intermediates. And so this builds the final virtual synthesized compounds. So it's important to clarify that our AI can go up to 4 steps, meaning that these steps can be repeated 3 more times. So the message here is that this is an in silico synthesis planning, but with chemical intuition built in. So our GenAI doesn't just propose ideas, it also proposes how to make them, which is very valuable, is very important if you want to apply real chemistry for your projects. So after discussing in silico experimentation and how we tackle synthesizability issues, now it's also important to discuss how we actually optimize the generated molecules towards your project's objectives. So at the heart of our GenAI is optimization using reinforcement learning in order to incrementally converge towards good molecules that are predicted to have strong properties. So why reward functions are important? Because they are essential to detect undesirable properties early. They are important to guide the AI in refining structure, prioritizing candidates, and enable effective multi-parametric optimization. So the possible rewards function that you can find in our platform using our AI is, for example, the Tanimoto similarity. So let's say that you have a chemical series of data sets, or maybe you have a design, a chemical structure of your design, and you want to explore around these structures, but you want to stay close to the chemical scaffold that you have, then you can use this Tanimoto similarity in order to optimize molecules. Then if you have the 3D structure of your protein and your reference ligand, then you can use our 3D structure-based rewards, which are the docking score and the interaction fingerprints. So here the interaction fingerprints is a very important reward because you can prioritize all those interactions that are absolutely important for you and that you want to see over other interactions. And so in this way, the algorithm will generate molecules that are optimized to show you these kind of interactions. So then if you have in-house models that you want to use to score your molecules and optimize them, we also have the external score that they can be easily connected to our platform. Then also we have in-house local models, like in our platform. Let's say that you have data for ARMI or activity, then you can build these models in our platform and you can use it to generate molecules, and these molecules, they're going to maximize the targets that you want to work with. And then finally, let's say that you don't have the structure of your protein, but you do have the information about your ligand, then you can use the 3D ligand base. And here what we do is we optimize the 3D shape similarity and the pharmacophore. So here I want to mention an important point, that all these reward functions for the multi-parametric optimization are fully based on user-defined rewards. It is you, the one that is going to decide which reward function you want to use for your project. So the decision and the upper hand on how you want to proceed is always on the user's end. So just building on the previous concept and just to close a bit the full image of our GenAI, once we define the target product profile, then the real works for the AI actually begins. Because we start with our generative AI, which designs novel chemical structures. But as I mentioned before, this is an unconstrained generation. It is grounded in real-world chemistry. The AI pulls from a library of over 1 million commercially available building blocks, more than 2,000 well-established organic reactions, and multiple reaction steps. So in other words, every molecule proposed is by design synthetically accessible. Then we enter the optimization loop. And these generated molecules that we had in the previous steps are now scored using the reinforcement learning based on how well they meet the target product profile criteria. So these scoring systems act like a reward function, guiding the AI to prioritize more promising candidates in every iteration. So the result is a tightly integrated, chemistry-aware design cycle that doesn't just generate ideas, it delivers optimized, synthesizable molecules and are aligned with your target product profile in your real-world projects. So I will give back the word to Quentin so you can discuss about the benchmark.
Thank you, Stephanie. So yeah, so we wanted actually to see how our GenAI perform compared to existing GenAI published. One very famous is Reinvent 4, published by AstraZeneca, which is very well maintained, which cover a lot of use case like growing, linking, etc. So it's really a good GenAI to compare with our system, because I believe that this Reinvent 4 is the best open source GenAI you can find. So it was a very good algorithm to compare with our Iktos GenAI. So we say, okay, let's take complex targets, synthetically difficult, with some interesting motif like macrocycles or complex side chains or complex central scaffold. And we try to ask the GenAI to be able to find those molecules back using a simple Tanimoto similarity. So we tried three different use cases. The first one is we asked those GenAI to find those molecules back without any information of the starting point. So the GenAI starts from nowhere and he has to understand by himself using a simple Tanimoto similarity to find the final molecules. As you may understand, a similarity of one means that the GenAI has been able to recover the molecules. Another use case was given a starting fragment and starting from here, try to grow the molecules to find back the expected molecules. And the final use case was a fragment linking. You give one fragment on the right, one fragment on the left, and you try to see if you can find the molecules. As you can see against all those molecules I just showed before, uh, Makia outperform reinvent in all those tasks. We have been able to retrieve the final molecules very often, and even in very complex tasks, for instance, with lorlatinib, which is a macrocycle, reinvent is not able to generate microcycles, so it stays very low in terms of Tanimoto similarity, but Makia has been able to understand the sequence of action with the final macrocyclization, which gives a Tanimoto of 1. So it's very interesting to see this type of behavior. Again, when you navigate into this chemical space of reaction, you make bigger jump and you can converge faster. So it's much more sample efficient. We tried another use case where we wanted to see in case of lead optimization. So we took a data set of 463 molecules published by Boehringer Ingelheim and measured on PI3K and mTOR, which are two kinases. We built QSAR models and we asked the GenAI, can you maximize the QED, which is the estimated drug likeness, the similarity of the initial data set. We wanted as well to maximize two QSAR models. We wanted to keep as well the main scaffold of that series, like in a traditional use case when the chemists want to keep some part of the molecules. And in post-processing, for the top 500 molecules, we compute the synthetic feasibility score, which is a retrosynthesis score. It's a true retrosynthesis. It's not an heuristic. We have published how we apply R-score by computing retrosynthesis. So in Makia, basically, we ask to start from this chloro and to grow in that direction. And in reinvent, you can ask the same. Of course, in reinvent, you don't have the synthetic feasibility notion. So you just put a star here and it will start growing from here. And what we saw is that the top 500 molecules against this multi-parametric fitness function, we compare the synthetic feasibility and we saw that Iktos GenAI produced 95% of molecules with a very good retrosynthetic feasibility score. So in 85% of the case, Iktos molecules are feasible, contrary to reinvent where it's in 50% of the case. On top of that, when you count the scaffold, so more or less the diversity, you can see that in reinvent, you have only 29 different scaffold compared to Iktos, where we have 81 scaffold. If you also compare scaffold similarity pairwise similarity, you can see that you have high similarity and a lower similarity, showing that we find more scaffold and more diverse. And the same if you compare molecular similarity, we have a lower similarity compared to reinvent. Again, another very interesting use case is to use structure-based information. So in that case, let's say that we are in a discovery phase, and we want to use structure-based information. So in that case, we took three different type of targets: PIM1, ARC2, TMRD, and we wanted to maximize a docking score. In that case, we use Autodoc VINA, but also a contact score, which is, as Stephanie explained, fingerprint interaction fingerprint. So basically, you can ask to the system that you want to interact with key amino acid in the pocket. For those three projects, we decided to use fragment growing. So we start from one exit vector and ask to generate in that direction from bromine here and boronic acid in this position. Again, maximizing a reward function. In reinvent, you just put a star here, here, and here, and reinvent as well is able to grow. What is very interesting is that on the PIM1 use case, if you take the top 500 molecules with the best docking score, in PIM1, reinvent works pretty well, and we obtain similar synthetic feasibility. In ARC2, we see a very high drop in terms of synthetic feasibility, because out of the 500 top molecules, only 99, so almost 100, were feasible. And it's a bit better on TRMD, but still half of the molecules are not feasible. So in most of the case, we are able to generate very feasible molecules. If we focus on the ARC2 example, where we start from this pyridine here, so of course you see the pyridine everywhere because it is the starting point we ask. As you can see, we have different type of scaffold and families of branches. And as you can see, very nice score, 0.90, 0.972, 0.964, etc., etc. So very good score. And if you take the best reinvent molecules, as you can see, the score is lower, 0.852. But also we get these crazy microcycles, which are almost impossible to make and not very drug-like. If you compare the diversity again on the three use cases, we have much more diversity because we have a lower pairwise Tanimoto similarity and also higher number of scaffold. And if you apply a t-SNE here, we can see that we explore a much bigger chemical space compared to Raven. So again, when you play with smiles during your generation, you play in small transformation, you play in character permutation, and it's different where you play with reaction because you are able to make bigger jump into the chemical space because you play with building blocks and reaction. And in all our benchmark, we can clearly see the difference between a sequence-based GenAI versus a reaction-based GenAI.
So I take back the control. So now, after we explained how we applied our GenAI for enable chemists in order to explore or experiment with in silico generation, how we tackle synthesizability, and also how our GenAI compares with others that you can find in the market. So now let's put everything together. And so let's talk now a bit more about the chemistry-guided de novo design of molecules and their optimization using these reward functions. So just for you to have an idea, our GenAI isn't limited to a single task. So it's built to handle a wide range of drug design challenges. So from scaffold hopping and heat expansion to lead optimization with multi-parametric optimization, we cover a really full spectrum, and all while ensuring synthetic accessibility, 3D constraints, and multi-parametric optimization align with your target product profile. So this is a real modular, I would say, flexible platform, which is designed to address your real world constraints in order to generate molecules and also to deliver viable candidates, I would say. So using our technology, you can have access to multiple paths to generate molecules. So if we start from this reference molecule, this PIM1 reference molecule here, for example, then we want to generate novel ideas from different starting points, then we can do it using our platform. So the first one is that maybe you can grow or you can want to generate new ideas starting from an initial fragment in one direction. So that's possible to do it with our platform. And here I show you an example. So here we are utilizing the bisphorophenyl with the boronic acid as the reactive center. So in this way, our GenAI will keep the scaffold, as you can see in all the molecules in this example, and then we'll introduce diversity in the rest of the molecules. Also, these molecules, they were generated with the 3D structure-based constraints as reward, which are the docking and context code. So the second thing that you can do with our algorithm is to grow molecules again, but let's say that in this case, you want to grow into different reactions. So you can do so as well. So here we have our core molecule with the amine and the boronic esters as reactive centers. So what we are asking here to our platform is to grow these molecules and to generate diversity, keeping this core in all the molecules and then generate diversity around this as well. Then the third use case that you can use with our platform, instead of working with a single initial fragment, now you have two of them and you want to bridge them, generating diversity in between. And so here is where you can perform, for example, scaffold hopping. So here in this example, I show you again, here we have the two different initial fragments, and then with our platform, we will introduce diversity in between the two of them. So then let's say that you're interested about macrocyclization and all you want to obtain macrocycles as your final results. This is also possible using our software. So here we have our starting point, which is the bromine and the chlorine. And what we're going to ask to our platform is to generate molecules, like kind of linking these two endpoints of the molecule and to give us macrocycle. In this case, we use another reward function, which is the 3D shape similarity and pharmacophore in order to get our final results. And as you can see, in all the cases, we are getting macrocycles. And so finally, the last use case that I want to talk regarding to our technology is that you can also fine tune. This is more used in the lead optimization stages where you have already a chemical series, which is defined and you already know which is the chemical scaffold that you want to explore. But you want to maybe increase activity or maybe increase ARMI properties, staying close to your chemical space. So this is also possible to do with our platform. So as you can see here with our technology, we can start from a common scaffold of your chemical series, and then we're gonna ask our software to give small modifications in the rest of the molecule in order to increase this reward. In this specific case, we work with QSAR models and similarity as reward functions to make sure we optimize molecules that maximize activity and ARMI models, and we stay also close to this initial chemical space. So to wrap it up, as I mentioned earlier, our platform is unique in the way that it leverages AI driven by chemistry and guided by chemists, which is very important in order to make synthesizable compounds. So on one side, we have our GenAI, which performs the de novo design of synthesizable molecules. And on the other side, the AI driven retrosynthesis technology finds the best synthetic route. As we discussed before, our approach to generative AI goes beyond simply generating molecules. Our target is to generate synthesizable molecules to actually accelerate the DMTA cycle. So to do so, we work with the datasets of, as I mentioned before, 1 million building blocks and 2,000 NEM reactions, incorporating chemistry into our GenAI technology. So we leverage the power of chemistry information to generate synthesizable molecules by design. And then these molecules, they can be optimized against the rewards that we discuss. And I want to insist again in the user-defined part, because at Iktos, we strongly believe that AI will not replace chemists. Actually, human expertise is essential to define your project's requirement, to choose a strong setup, and also eventually select the interesting results to further analysis. Okay, so, and finally, you can use our AI-driven photosynthesis software in order to help the chemist prioritize those molecules that are easier to make, maybe, and also to meet the requirements by the medicinal chemist with respect to cost, time, and complexity optimization as well. So I will give back the word to Quentin so we can discuss the use case. I'm going to mute myself and then I will give you control, Quentin.
Thank you,
I
Stephanie.
cannot.
So
Okay.
yeah, I'm going through, the application of this technology in a very crowded IP space and showing you how in 3 months we have been able to find new chemical matter. So it was a PKMIT1 use cases. It's kinase coming from the We family, and it regulates CDK1, so it has an important role in DNA damage response. And when you go to the literatures, you can see that there is one compound which has been designed by Repair Therapeutics, which is its phase one, right now, sorry. And it's a very potent molecules you can see here in the PKMIT1 pocket. It's very interesting because this type of molecules contain very nice features, which is this NH2 in this position. Also, you have the phenol, which sits in the back pocket, and here the amide function, which interact with the hinge binding motif. And Reaper explore lots in that chemical space, always keeping this NH2, this phenol, and this amine moiety on the left-hand side, with some small modification, but always those three features. When you see the followers in that field, it's the same. Everyone is designing either a phenol, or I would say something similar, having similar interaction, plus NH2, plus amine moiety. And you always see this type of motif everywhere, even at in silico, with one also compound with a tiny, which is a bit different, which is interesting to observe that at in silico medicine. But in most of the case, we see always this type of features. So we say, okay, can we use Machia? Can we use our retrosynthesis technology, and on top of that, our robot of chemistry, to be able to discover something which is different from this classic approach of NH2 amide on that part of the molecules? So what we decided, as Stephanie explained, we decided to start from the back pocket and to keep the phenol, which seems to be an important features, and trying to grow in that direction to identify something completely different, completely new. So the fitness function in that case was reactivity. So we define either bromine, either boronic acid, either with a dimethyl phenol or monomethyl phenol. And we wanted to maximize QED, which is estimated drug likeness. At the same time, we wanted to have a very good structure-based score, making key interaction into the pocket, again, defined by the ComChem and the MedChem at Iktos. And then we have been able to generate 30 totally new scaffold, and we decided to synthesize 16 molecules. And these molecules has been synthesized on our robotic platform. We identify 4 active scaffold never published before, with the absence of this NH2 and this kind of amide function. So as you can see, with very nice activity, 30 nanomolar activity, 200 nanomolar, 600 nanomolar, and 267 nanomolar. Unfortunately, I cannot show you the design, but you can trust me that it's a totally novel scaffold never described before. We tried another experiment, keeping the amine moiety on the right-hand side and keeping the phenol moiety on the left-hand side. Again, with this type of reactivity for the phenol and this type of the reactivity for the amine moiety, trying again to find a new scaffold in the middle. We synthesize 24 molecules. We identify 3 active scaffolds, a bit less active than the previous campaign, but still interesting starting point with a completely new scaffold with, again, the absence of this NH2 moiety.
Yeah.
Stephanie,
Yes, of course. So this is just the last use case that we wanted to showcase, because the last one, it was just like a project where unfortunately where we cannot show any kind of results, but this is more a use case on how you can actually use our platform for different kind of targets. In this case, I'm gonna explore the GLP-1R targets. So so this use case, the first thing that I want to say is like it highlights the kind of problem solving that you can achieve in Makia when working with a challenging targets, like in this case, the GLP-1R transmembrane protein. So GLP-1R is a particularly important target because when activated by its natural agonist, the GLP-1, I'm sorry, it triggers a G protein signaling cascade that actually leads to insulin release and glucagon inhibition. So this makes a key targets in treatments of the specific disease, like for example, type 2 diabetes. And it's also a very challenging target per se, because it's a transmembrane one. So there are several peptide-based GLP-1 agonists already on the market. However, there is a growing need of new small molecule agonists. In this use case, we decided to work with the Pfizer's compound Perron, which was recently discontinued. However, it's shown promising results, and we can use it as starting point to design new chemical scaffolds. So the initial success of Perron can be largely attributed to its ability to engage deep within the receptor binding pockets, specifically through quantum bridging interactions with residues which are buried in the transmembrane region of the GLP-1R through its carboxylic acid here at this position. Okay, so this critical interaction helps actually stabilize the protein molecule complex, effectively mimicking contacts made by the natural agonist. Then on the other hand, the oxidane moiety here forms important hydrogen bonds that contribute significantly to the compound's potency. So with this in mind, that if we want to use all this information to build a use case using our platform, if we want to design this novel compound starting from this reference ligand, it is crucial to retain both the carboxylic acid and the oxidane as starting points. So we applied a fragment-based approach to generate novel molecules using these key functional groups as core elements. So through fragment growing algorithm, as the one that we've shown before, we will introduce chemical diversity while preserving critical interactions within the binding pockets. So starting from the previous initial fragments, we target the generation of novel chemistry, and so we keep the crucial part of the molecules, and then we want to generate diversity in other areas. So this strategy allowed us to explore interactions that are already there, like the ones that I showed you here in the left, and also to explore new interactions with residues like leucine 217, which is, it would be a new interaction. The ligand does not feature this one, and will allow to generate a more diverse set of molecules. So now that we have defined our project goals and we have identified the initial fragments, it is possible to start with the generation. So from our software perspective, we have our initial fragment, which contain the oxidane and the carboxylic acid. And then what we want to do is to grow starting from here, as explained before. So then from the point of view of reward functions, we use two of them, which are local models, activity models, and then we have also the structure base. And importantly, we use these two reward functions at the same time in the same generator. So this is also important to understand, that our platform allows you to perform multi-parametric optimizations in different kind of rewards. You don't have to work with one reward and then with the other. You can couple them at the same time. So from the structure-based point of view, we also targeted interactions that we were interested in, like as I mentioned before, like the leucine 217, in order to guide the generation into that direction. So at this point, and I think Elle will share you, and I think it's important if you can go into this page, because here it's a demo page where we are gonna give you a full demonstration of the GLP-1 use case case, how you can set up the parameters using our software, which is Makia, how you can actually target the interactions, how you can use the 3D structure base. We're going to show you the results and how we prioritize them as well afterwards with our Spaya, which is the AI-driven photosynthesis software. So here you have the code to scan, and I think Ele will share it as well in the chat. So I invite you to go and take a look. You will also find demo videos of another use cases, publications, and different kind of usual questions that we always received. And so finally, the very last thing, and before just running out of time for your question, the last thing that we wanted just to share with you is that it's very important because, of course, you can design your molecule, you can generate them, but then you would ask, okay, how can I do using a software to prioritize them, and how can I actually visualize everything? And so we're really working on a new version of our software, which is Makia 2.0. And here the idea is that we will give you the upper hand in order for you to perform all the analysis of the results. So here I just have a screenshot again, and what we are hoping is, apart from being able to generate molecules, is also to give you tools in order to analyze your results. As you see here in this screenshot, you will have different kind of ways to explore your results and to prioritize the molecules that you want to keep. So once again, I invite you to our demo page, because there you will be able to visualize a small video in where you'll have a preview of what's about to come with respect to our software. And so with this, first of all, before jumping into the Q&A, I want to thank you all for your time and for being here. We are very excited and very happy to share everything that we are able to do at Iktos. And so I will give the word back to Ele once again.
Thank you very much, Stephanie, Quentin, for the presentation. Really interesting. So as you mentioned, we will jump right into the Q&A as questions have been flowing. So I will start reading a question from Yulia. Are you able to share more details on the 6 million reactions that you trained the model on? Are these public data or generated internally?
Yeah, so the reaction we are using are reaction coming from the patent literatures. So it's public database, and it's a 6 million reaction, yes. And everything which has been published can be used during the GenAI. So I take the opportunity also to answer to another question related to the diversity of the reaction, because we are not doing only cross-coupling. We are doing any type of published transformation into the patterns. So we can do cyclization, macrocyclization, heterocyclic chemistry, everything you can imagine. For instance, Stephanie, the GLP-1 use cases to form the scaffold. And you can see that by following the QR code and the link proposed by Eleonora. You can see which type of transformation we use to start growing from the starting points.
Thanks, Quentin, for the answer. So related follow-up of the building blocks, I will read a question from Christopher. He asked, you mentioned that your models rely on a curated CAB database of 1 million building blocks. How easy can a user integrate their own custom building block library into the generation process? Is this as simple as providing a new set of fingerprints for the similarity search, or is a significant model fine-tuning required to optimize performance with a new chemical space?
So there is no retraining, and it's very easy to add. A lot of clients ask for these features because, of course, they have in-house inventory and they want to take advantage of that. And so it's very easy. You just share a list of smiles and we can integrate it to Makia. And the GenAI will try to predict that directly from this database using fingerprints, as you mentioned, but no need to retrain it.
Thanks, Quentin. Also related question that you answered partially. Is the generation fully based on existing or virtual building blocks? And what if an ideal motif does not exist in the building block template library?
So it can be any type of building blocks. It can be existing one. You can also triage those one by cheap one or more expensive one, more exotic one. It can be virtual one. You can really define the one you want. And if by if it happens that you don't get the building blocks needed, don't forget that Micah is able to apply chemistry on the generated molecule. So it's not only addition of building blocks, but it's also chemistry without addition of building blocks, because we have all the published transformation. So you can have oxidation, reduction, any type of transformation, functional group interconversion. So even if your building blocks, ideal building blocks, is not present, it can be generated starting from a simple one. You're on mute, Eleonora, or I don't hear you anymore. I don't know if it's coming from my
Yeah,
side.
I don't hear you anymore either.
No, we lost you. Maybe I can take over. How fast is the structure-based reward? Is it only used in the final scoring step, or it is used iteratively during the generation? So that's a good question. Structure-based reward can take time. That's why in Micah we are on AWS. It's a parallelized platform. We can dock at the same times thousands of molecules, because at each step of generative AI we generate 1,000 molecules. So the GenAI, as Stephanie showed, combine reaction and building blocks to virtually generate the molecules. Those molecules, so at each batch, 1,000 molecules are sent to the docking score. We score them, and then we feed back this score to generate a new batch. So it's not a post-processing filtration. It's a score which is taken batch after batch during the generation to maximize and to climb the fitness function along the iteration and the batch.
Thanks, Quentin. Can you hear me now?
Yes.
Yes,
Okay,
we
great.
can.
Because as you're discussing scoring molecules, we have a question that I think comes from when you were discussing the benchmark study. So Jenna asked, how was the molecule synthesizability calculated, and what does the R score of over 0.5 really means?
Stephanie, you want to take it, or?
I can explain the R score and you can explain the how you can calculate the synthesizability. So the R score that we have, it's actually the first thing that it's important to clarify is that it's a real R score because we compute it. So we do not predict anything. So we have different kind of ways that we computed the components for this computation of the score. Like, for example, the feasibility of the disconnection, how similar it is to certain disconnections that we know already in the patent, the applicability domain, also the number of steps, because of course you don't want a feasible route with 15 steps. That's not very useful. So it takes into account all these kind of things just to compute the R score. So with this being said, R score, it goes from 0 to 1. So 0, it means that the molecule is absolutely, according to the algorithm, not synthesizable. And 1, it means that it found exactly that transformation in one step. So 0.5, in our opinion, it's already a good balance, because we don't want to say 0.8 or 0.9, because also we want to give the chemist the opportunity to revise the tree, and with a R score of 0.5. And so yes, that's what it's the meaning of the number, and how we actually compute the score.
Yeah, it's very important to understand that heuristics are not working. If you use the synthetic complexity score, if you use the synthetic accessibility score, if you use the RS score published by AstraZeneca, there are prediction. So it takes a smiles, it predicts a score, and it's very easy to hack those type of scores, because you just move one bounds and you transform a very feasible molecules to something totally unfeasible. And those type of score are not able to see that. So the R score is a real retrosynthesis. So it takes time. It takes between 5 seconds to 20 seconds. But at the end, you have very meaningful scores.
And if I may add, in this demo page, you will find a publication that we have related with the art school, which is very interesting, and we compare it with the other heuristic methods that Quentin mentioned before.
And if you want to see how it looks, Spaya is publicly available on
Absolutely.
spaya.ai, so you can
Yeah.
create an account and put your molecules, and you will be able to see the different routes and the different scores associated to it.
Thanks both for the answer. I think we have an interesting questions that relates to the chemical space that you mentioned, Quentin, our assessments of the 10 to the 27. The question is from Andrea. He asked, as you mentioned that the Iktos space is 10 to the 27, can you please explain what kind of medicinal chemistry rules are used and how much curation is done for that space?
So that space is not created because it does not exist, because you cannot put somewhere in a database 10 to the power of 27 molecules. So it's a chemical space that we can theoretically explore, as Stephanie explained, by using 1 million building blocks, 2,000 name reaction, and combining that over 4 steps, we theoretically explore 10 to the power of 27 molecules. And then when we generate those molecules, theoretically they are feasible, but at the end of the day, you will really want to make sure that you can make it to the lab. That's why Stephanie presented that in backend, we have also Spaya, which is here to perform retrosynthesis on all those generating molecules. So at the end of the day, you don't need to pre-compute this library. You don't need to curate it, because by design, we generate feasible molecules. And on top of that, we have retrosynthesis behind.
Thank you. We are on time, but we can go a bit 2 more minutes so we can answer all the questions that we have on the chat. A question is for Udin. He asked, can GenAI give synthesis step prediction for flow continuous reaction? There is different synthesizable criteria for batch reactions and flow continuous reactions?
So, it's Spaya and Micah are totally agnostic on the way of assessing the chemistry, if it's in batch or flow. So the answer is yes, you can take either batch, either flow chemistry. Of course, when you will go to the lab, the setup will be different. But if Spaya find routes passing by a Suzuki coupling and heterocyclization, if those reactions are amenable in flow chemistry or in batch chemistry, the output is the same, it's feasible.
Thank you for that. And then we wanted to address some questions from John. You answered partially, but our attendees cannot see the questions asked. So I will read the question around chirality. For chiral target compounds, it appears that your chemical transformation algorithm will rely on building blocks that already contain the stereochemistry information versus leveraging asymmetric transformations to set the stereocenters in the target molecule. Can you please verify if this is the case? And if so, how would you see improving the algorithm to include asymmetric transformations in synthetic planning?
Yeah, that's a very good question. Asymmetric transformation is very tricky because it's come from also, again, Spaya, Micah are data-driven technology. So it used database to learn chemistry. And right now in public database, asymmetric transformation are still difficult to exploit, because sometimes you have chirality which is not well defined. Sometimes it's chiral, but actually you have a racemate. So it's very difficult to handle asymmetric transformation. So right now, as you spotted, we use chirality information, but the stereocenters is already pre-installed in the building blocks. And this is for sure something we plan to change in the futures, not only for Mica, but also for retrosynthesis, because we believe that being able to provide a chiral transformation in retrosynthesis will be a very cool feature.
Thank you. And I think we have our last question from Udin. He asked, we have experience, we use docking to develop new chemical entities for COVID-19 drugs, but after synthesis, we know there were problems with solubility in the actual lab. So there might be a need to develop salts or esters to improve solubility. how we can make sure that the compounds generated from the generative AI will give better physical chemical properties for drugs, not only around synthesizable score.
Yeah, that's a very good question. And this was the first slide of the webinar. Of course, synthetic feasibility, it's very important and very difficult to tackle. You know, we spent several years developing this type of algorithm, but it's not only the only reward we are adding to the fitness function. As Stephanie showed, we can use shape, we can use pharmacophore, we can use docking, we can use descriptors, we can use QSAR models, global, local, on solubility, on stability, etc., etc. And this is something we do on a daily basis at Iktos, and also all our clients using Micah can put this type of constraints. And just to give you an example, here in the PKMIT one, we performed some solubility test, and the compound were all soluble because we have put into the system this type of descriptors and fitness function.